देखो, तो अभी जो हम लोग टॉपिक देखते जा रहे हैं वो क्या हम लोगों का डेवलपमेंट ऑफ एम्ब्रायो। ठीक है, इनसाइड डेवलपमेंट ऑफ एम्ब्रायो द फर्स्ट विच विल बी सीइंग इज योर डाइकॉट। ठीक है, और अगर हम डाइकॉट का अंदर बात करें तो हम लोग क्या देखते हैं, इनिशियली हमारे पास क्या होगा, विल हैव अ जाइगोट। ओके, ये हमारा जाइगोट हो गया, डिप्लॉयड सेल है। नाउ दिस जाइगोट, इफ वी आर लुकिंग एट व्हाट वी कैन सी हियर इज कि दिस जाइगोट, this side we can say it is a micropyle end, and this side we can say it is a chalager end. So this zygote will divide into two different type of cell. Just to differentiate between the cells, we'll change the color of the cells. So it is divided into two different type of cells. The cell towards the chalager end is known as the terminal cell. The cell towards the particular micropyle end will be known as the basal cell. Now what will happen if we look here, what do we see here is this terminal cell will divide to give a globular head. Okay, so this terminal cell will divide to form a globular head which is now containing 4 different cells. Now on the other hand, this basal cell will divide to give suspensor. Now if we are looking at suspensor, what do we have? We have numerous cell here, 1 2, like this many cells will be there here. And if we are talking about the cells, what do we see here is the last cell will be a bit larger in size. So if we look at the entire group of cell, this is called suspensor. If we are talking about the last cell which is here, it is called hostria. The reason it is called hostria, because it push this particular globular head into the endosperm region. Okay, it will push it into endosperm region, because of which this particular globular head will get the nutrition to survive. Okay, so initially we get a 4 cell globular head. Later the same 4 cell globular head will again divide to give us a 8 cell globular head. So this will give us 8 cell globular head. And if we are looking at the particular first cell, which was this one, we will change the color of this cell just because this cell will perform a specific function. We'll talk about this cell. Below this we will find other cells which are normal, just like the first one. And below this, let us have the suspensor here, the ostial cell here. So this cell which we have drawn in blue color is basically known as hypophysis. ओके, इसको हम लोग क्या नाम से जानते हैं सर? इसको हम लोग हाइपोफाइसिस के नाम से जानते हैं. Now if we are talking about the hypophysis cell, what do we see here is this hypophysis cell will divide to form a 8 cell stage, and at the same time this globular head will also divide to form a 16 cell stage. So if we are looking at our head, what do we see here is this is the 8 cell stage which we had from before. This time the division will be taking place in such a fashion that we get 2 layers of cells. So we'll have basically the 2 layers of cell which will be forming here. So this is how the 2 layer of cell will form. Now this kind of division which lead to the formation of 2 layer of cell is known as our periclinal division. Periclinal division. Okay, now what we have said, that the hypophysis cell will also divide, and it will divide to give us a 8 cell stage. So this is our 8 cell hypophysis stage, below which we will have those other normal cells which we had before also. So these are the other normal cells of the suspensor. Okay, now these three stages which we have represents our globular endosperm. It represents our globular embryo. Now, talking about this globular embryo, what do we see here is, we can divide this globular embryo we can divide this globular embryo into two halves. One is the upper half, one is the lower half. So what do we write here is, here the upper half, if we are talking about, this upper half will give rise to, if we are talking about the upper half, what the upper half will form? It will give rise to the epicotyle region plus the cotyledon which we have in the seed. If we are talking about the lower half, it give rise to the particular hypocotyle. Okay, it will give rise to what? It will give rise to the hypocotyle region. See here, if we are talking about these cells, the upper four cells, if we are talking about, this will give rise to the root cortex, and this here will give rise to the root cap. So if we are looking at the globular embryo, we can differentiate which region will give rise to which particular cell. So we have seen that the upper half will give rise to the particular epicotyle and cotyledon, the lower half will give rise to hypocotyle, the four hypophysis cell near the globular head will give rise to root cortex, and the four hypophysis cell below will give rise to root cap. Now this globular head will further divide. Okay, this globular head will further divide, and what it will do? It will form a heart shape. What it will do is, it will form a heart shape embryo. So what we will get now is a heart shaped embryo which will be present something like this. Okay, so this represents your heart shaped embryo. Here we'll have the epicotyle. Now what will happen? This portion and this portion will divide further. Okay, and this will lead to the formation of your cotyledon. So what we're gonna get here is this structure. This is the suspensor region which is present below with the haustral cell. Now here will be the epicotyle which will be present. So this structure after the heart-shaped embryo is known as horseshoe embryo. So if we are talking about development of embryo in dicot, what do we basically see? We have three different stages of embryo: globular embryo, heart-shaped embryo, horseshoe-shaped embryo. I'll tell you which are the questions which can be asked. Okay, so they will be asking what is the sequence of the embryo: globular, heart-shaped, horseshoe-shaped. In globular embryo, they can also ask the sequence. So you should say 4-cell stage, 8-cell stage, and 16-cell stage. Clear? So these are the questions which can be asked from the development of dicots. Clear, Babu?